In this last video for Chapter 4, we want to talk about some of the principles and ideas that we would use for classifying and identifying unknown microorganisms. Classification of microorganisms is sometimes challenging because, as we've started to see in the laboratory section, many microorganisms look extremely similar to one another. However, I think we all probably have a good idea, a good sense that different microorganisms are responsible for causing different kinds of diseases when they get into the human body. And so being able to identify an unknown microorganism can help us very quickly to be able to figure out what the disease is that the patient might have, and consequently be able to lead to an appropriate treatment for that patient. Scientists, biologists in particular, love to be able to classify microorganisms from a broad spectrum, broad perspective. It helps us to get a sense of the order of different living microorganisms. Amongst scientists, it helps us to be able to enhance the communication of what we found, our results and interpretations from one another. Classification of organisms can lead us to be able to make predictions about structures and functions, which from a clinical standpoint can help us to lead to perhaps better therapies. And then finally, the science of biology is held together by the broad principle of evolutionary relatedness of all living systems, and the investigation and classification of living cells can help us to be able to draw this sort of evolutionary connections. The first big classification scheme that was really put forward by biologists was by Carl Linnaeus, who initially classified all living systems according to two kingdoms, which were based on their morphological or appearance types of characteristics. And he broadly called these things plants and animals. And if you were a living system, a living organism, you fell into one of those two camps. Now, when Linnaeus was busy classifying organisms, he was aware of microorganisms, but they didn't particularly fit into his classification scheme. That really didn't begin to fall into play until about the 20th century, when Robert Whittaker proposed the five kingdoms that were really the basis of textbooks in biology all through the 20th century. The five kingdoms of Whittaker were broadly classified as plants, animals, fungi, which were all multicellular in nature and were differentiated based on the way that the individual kingdom was able to obtain its nutrition. Plants, of course, are photosynthetic and get their nutrition from the light of the sun. Animals are motile organisms, movable organisms that go out and catch and ingest and digest their food. Fungi were kind of an intermediate and stationary, closely resemble plants, but of course are unable to get their nutrition from the light of the sun. They're not photosynthetic. They actually secrete enzymes and digest the food out in the environment and bring up the food back into their bodies. Whittaker also recognized two unicellular kingdoms, the protista, which were composed of eukaryotic cells, and as we'll see when we talk about eukaryotes in chapter 12, the protista are an incredibly broad range of organisms. Some of them are photosynthetic and some of them are not. And then finally, Whittaker recognized that the prokaryotes were very different in cellular structure, and these were given their own kingdom. So Linnaeus's goal when he was beginning to classify these things was simply to begin to try to catalog all the different microorganisms or all the different organisms that he was able to view. All of his classification schemes was really based upon the way things appeared. And so two organisms that had hair and were warm-blooded and had live young, he classified them as being related to one another simply because of the types of bodies that those organisms have. Today, the more modern goal of understanding these taxonomic relationships is to really begin to describe these relationships in more detail. And the major goal of what we try to do as we classify organisms today is to really begin to reflect that relatedness of different organisms to one another. And as a consequence, what we find is that the physical appearance of organisms sometimes can be a bit of a red herring. And we can have two organisms that may appear to resemble one another from a morphological standpoint, the way that they appear. But when we examine their genetic material, that has leads us to think that perhaps just appearance is not sufficient for being able to help us to classify and the relatedness of organisms. The emphasis on the genetic material and the analysis and comparison of the genetic material has led us to add an additional domain to living systems. And this has really been championed by the microbiologist Carl Weiss, who took it upon himself actually to begin to examine the DNA sequences of different microorganisms. And the DNA sequence that he chose to examine was actually a DNA sequence that encodes a component of the ribosome. We recall from chapter 3 that all living cells, prokaryote and eukaryote, have ribosomes, and those ribosomes do essentially the same job in all living cells. They're a participant in the process of protein synthesis. For an organelle that's found in all living cells, what this means is that it's not able to mutate at a particularly high rate. Mutations, of course, happen in ribosomes, but a detrimental mutation is going to make that ribosome so it just doesn't work and will not appear in nature. Ribosomal sequences, as a result, change very slowly and very rarely, and so it can be used as a very useful yardstick to be able to measure the differences between different kinds of microorganisms. What we do then is we sequence the DNA of the gene that encodes these ribosomal genes, and we compare them by lining the DNA sequences up one against the other. Two DNA sequences from different organisms that are extremely similar, we would say that those two organisms are very, very related to one another. If we look at those same DNA sequences from two other organisms and we find that they are very different, we would say that those two organisms are not related to one another. And so Weiss did this. He did this with many, many, many different microorganisms. And he did this with also some animals and plants as well. After completing this, what he then proposed was, instead of 5 kingdoms as we saw in the previous slide, he was proposing 3 domains of life, which were based on 3 basic types of cells, as determined by comparing these ribosomal DNA sequences. The 3 domains that Weiss proposed were eukarya, which is composed of all eukaryotic organisms, plants, animals, fungi, protista, and 2 classes of prokaryotic organisms, the bacteria and the archaea. And what Weiss found was that representatives from each of these different domains were easily comparable and showed similar genetic characteristics to one another, which really set them apart in these 3 separate domains. So to step back just a little bit and bring this back into the clinical microbiology laboratory, let's talk about some strategies then that we can use to try to classify microorganisms, and if we have an unknown microorganism, to talk about strategies that we might want to be able to use in order to be able to identify what that unknown is. These are broadly listed right here in these bullet points right here. We can use the physical characteristics of a microorganism, the morphology, the appearance under the microscope, the shape of the cells, and that can give us an important clue as to what a microorganism is. We can also utilize biochemical tests, and we'll talk about a couple of examples and see how those are read and interpreted. We can use serological tests, which are going to be another approach for trying to identify a microorganism. It has an advantage that it's extremely rapid, but we'll also talk about one important disadvantage that serological tests have. We'll talk briefly about phage typing, but we're not going to have in our laboratory section to be able to have any experience with phage typing. It has an important basis in clinical laboratories, so we'll want to know at least the principle about it, but we may not be able to run and talk about it in huge detail in Bio 230. And then finally, we can analyze nucleic acids, which is really probably going to be the hallmark of the future for identifying microorganisms. So morphology, the appearance of a microorganism, we can be handed an unknown microorganism. We can stain it with a differential stain, with a simple stain, an acid dye, a basic dye, and we can put that slide under the microscope, and we can see microorganisms will have very, very different appearances from one another. The problem is that we sometimes have microorganisms that look extremely similar under the microscope, and yet are different species. An excellent example of this would be the bacteria Streptococcus pyogenes and Streptococcus pneumoniae. They're going to be almost indistinguishable under the microscope. It'd be take a very well trained microbiologist to be able to differentiate those under the microscope. And yet Streptococcus pyogenes and Streptococcus pneumoniae cause different diseases, as you might guess. So the morphology of microorganisms can give us an important clue as to what a microorganism is, but it's going to have its disadvantages. It's not going to be able to tell us precisely what the organism is down to the species level unless we're extremely lucky. Biochemical tests are going to be used extensively in the microbiology laboratory to help us to be able to identify microorganisms. The principle behind a biochemical test is this. A microorganism is given a substrate that an enzyme may perhaps be able to act upon, and a product is formed. When we see here are tubes that have a kind of broth, and we can see that over here in the tube on the left that the biochemical activities of the microorganism in this liquid has produced a bubble of gas. The different microorganism that's in this tube, the same type of broth media, no gas bubble is produced. And so we would read this tube as being positive for gas production, this tube as being negative for gas production. We can see that the third microorganism that's over in this tube has not changed the color of the media, and so consequently we would read this tube differently. The principle behind this can be analyzed or described perhaps a little bit more detail with this biochemical reaction where the sugar lactose can be broken down to produce gas, carbon dioxide. It's also going to be used to produce energy, but the carbon dioxide gas is what we see. A microorganism that has the enzymes necessary to be able to accomplish this reaction would produce the gas. A different microorganism that lacks the enzymes in order to be able to do this would give a very, very different result. And so we might interpret this tube over here on the left as being positive for the activity, the enzyme activity of taking lactose and producing gas from it. And this result can give us, again, a very important clue as to what the microorganism is. We have a different biochemical test over here. In the chemical test over here, the precursor cysteine is broken down into sulfide, and in the tube over here on the left, hydrogen sulfide has been produced. The tube over here on the right, no hydrogen sulfide has been produced. So this organism would be negative for sulfide production. This organism over here would be positive for sulfide production. We can use tests like this to be able to help us again to get very good clues as to what an unknown microorganism is. We'll see a general strategy that we can use in order to be able to use these biochemical tests to facilitate the identification. These are very nice. It's very obvious about the difference between this tube, and this tube, and over here the differences between these two tubes. The drawback of using biochemical tests, the problem with it, is that they all require incubation, which means that if you have an unknown microorganism on a microscope slide or that you've just pulled from a patient, you're not going to be able to get these answers until after a period of time that the microorganism has been able to grow sufficiently to produce the gas bubble or to produce the hydrogen sulfide. If you need an answer super quick, this is not going to give you the answer. We can use biochemical tests and run them many tests all at once, which can help us to get our answers somewhat more quickly. The devices that we see held in this scientist's fingers is a device called an interrotube, and you can see it's divided up into a bunch of different sections. Each of these different sections represents a different biochemical test, and what the scientist has done has been to inoculate each of these different chambers all at one time. And then the interrotube is incubated for a period of time, and then each of these little sections is read for either being a positive result or a negative result on down here. And we can see that these three different interrotubes with three different microorganisms can give a different pattern of results. So look over here at this fifth one over here. it's pink up here, but this microorganism is producing a yellow result. This organism is producing a pink result. What we can see is that these three different organisms are giving different patterns of positive and negative results for each of these. And so these interrotubes can be a very rapid way for trying to narrow down what an unknown microorganism is by running many biochemical tests at once. Again, though, the drawback is that even though you're able to run a dozen tests all at once, you're still going to have to wait for the positive tests in this interrotube to be able to grow sufficiently to give you the positive results. So again, these are going to require incubation, just like we saw before. Well, what if you're extremely impatient? You need to have a result extremely quickly for what a microorganism is. We can utilize serological tests. The one that's commonly used in microbiology laboratories all the time is a test known as the agglutination test. And the principle is a microorganism, such as these little blue spiky balls that we see right here, are mixed with a chemical called an antibody. Anytime we hear this word serological, what this means is it's a reagent from blood. It's an antibody that's been purified from blood. If the chemical reagent is able to react with an unknown microorganism, what we see with the agglutination test is a clumping reaction. We can see a clumping reactions occurred when this unknown microorganism's been mixed with our special antibody chemical. Over here on the right, it's a different microorganism. You can see that the little spiky blue balls look different from these, and consequently no clumping occurs. The great advantage of a serological test like this is that this positive result appears extremely rapidly within a matter of minutes after mixing it with an unknown microorganism. The most common one that you may have heard of that's used in clinical labs in doctors' offices all over the place is the rapid strep test. And if you've ever had one of these, what they do is they swab the back of your throat, and what they do is they simply mix it with an antibody reagent, and they're able to look for this extremely rapid clumping reaction that happens within a couple of minutes. They'll come back from the lab at the doctor's office and say, yeah, you got strep, so here's an antibiotic. The problem is, what organism do you have if you have a negative result? Let's say your throat hurts. You think you have strep throat. You go to the doctor's office and they swab your throat and they mix it with this chemical, and you end up with a negative result over here. What do you have if you don't have strep? Well, there's lots of things that you could potentially have if you don't have strep, and the problem with the negative result is that you still don't know what's wrong with the patient. In the case of the biochemical test that we saw previously, we still have some useful information that can facilitate the identification of an unknown. With a serological test, although it's extremely rapid, having a negative result doesn't really help us any closer to finding out what that is wrong with the patient. It simply tells us the patient doesn't have, for instance, strep throat. Phage typing is another technique that's used in the clinical laboratory, and it uses an unknown microorganism with a known virus. The viruses that infect bacteria are known as bacteriophages, or phage for short. When we talk about viruses, we'll talk a little bit more about the activity of bacteriophages when they're mixed with bacteria. But the end result is that when a bacteriophage is infecting a bacterium that it's able to infect, it's going to end up destroying that bacterium. And what's viewed when a phage is mixed with a bacterium is a clear zone of lysis, which means that the bacteriophage that was added right here in this circle was able to infect and destroy the bacteria on this plate. What's done with phage typing is an unknown microorganism is mixed with a bunch of different phages. And if we end up with a positive result, what that means is that we end up with this clear zone of lysis, which can help us to be able to identify what that unknown microorganism is. Because if the phage is able to infect that bacterium, it's not an unknown microorganism anymore. It's going to be one that's going to have a very distinct relationship with the bacterium. The problem is that if we set up a phage typing with a bunch of different known phages in an unknown bacterium and we observe no destruction at all, my bacterium is still unidentified. And so much like the serological test, if we end up with no positive results at all, we still don't know what our microorganism is. Well, let's talk about something that can really help us out. What's going to happen in clinical labs over the course of the next couple of years is that a lot of these techniques that we've seen in the previous couple of slides are going to become more and more obsolete. The new way that we can analyze unknown microorganisms is by analyzing DNA. The way that this is currently being done in clinical laboratories is a sample of DNA is isolated from an unknown microorganism. Through using the polymerase chain reaction, which you guys may remember from Biology 1, a specific gene sequence, a specific DNA sequence in this isolated DNA is amplified, and the gene of choice is usually the 16S ribosomal gene that we already talked about with Carl Weiss. It's the one that Carl Weiss was using. The reason that this gene sequence is used is because it's easy to amplify from pretty much any prokaryotic microorganism. From our isolated DNA, our PCR reaction, we have an amplified ribosomal gene, and it's a trivial matter today to be able to figure out the DNA sequence of this PCR product, and then that DNA sequence is dumped into a computer, and we analyze our unknown DNA sequence with the DNA database that's in the computer, and in about 15 seconds that unknown microorganism can be identified. This flow chart that's shown here is actually relatively rapid. We actually have used this procedure during summer microbiology during the laboratory section to be able to identify unknowns that we pull out of Tyler Run during the summer session. The isolation of the DNA takes probably about 30 minutes. The polymerase chain reaction takes about 2 hours. The actual time block that we have in here is figuring out the DNA sequence, because we don't have the capacity at York College to be able to determine the DNA sequence of an unknown microorganism. What we do is we put our polymerase chain reaction products into an envelope and mail them out to somebody who does do our DNA sequencing. When the PCR reaction is put into the DNA sequencer, it's about another 2 hours of time to determine the DNA sequence, and then once we get the DNA sequence, this comparison of the DNA sequence using the computer is about 15 seconds, and we identify our unknown. If we add up the time that we have here, we have 30 minutes, 2 hours, 15 seconds. We have a total amount of time of about 4.5 hours to go from having an unknown and identifying our unknown. And we can see that although this is slower than serology, it's faster than all of the other techniques in terms of the amount of time that's actually spent in the laboratory. And the other advantage is that there are no negative results. We have an answer of what our microorganism is here at the end. Now, it is possible that the unknown that we identify may be brand new to science, which would be awesome, because then we write up about the unknown that's made our patient sick and become very, very famous with a publication for it. But odds are, in a clinical laboratory that's going to be looking at microorganisms, unknown microorganisms that are making patients sick, what we're going to find is we're usually going to get a hit. that's going to only take a couple of hours. We'll be able to get that patient onto the appropriate treatment very quickly. Most identifications in clinical labs are not using this approach yet, but I anticipate that here in the next couple of years this is going to become much more frequent, simply because of the time savings that it involves, and it's also not a terribly expensive procedure to do this at all. And many clinical laboratories around the United States have all of this equipment in place to be able to move this along. All right, so let's talk about some of the strategies that we might use to identify an unknown microorganism in the Bio 230 lab. The big tool that we're going to use is something that's called a dichotomous key, which is going to be a series of yes or no questions that are worded so that if we have an unknown microorganism, we perform a test that has either a positive or a negative result, and based on whatever the result that we get with that test, it'll lead us to another test to do until after we finish this procedure we know what our unknown microorganism is. Here's an example of a dichotomous key, and we can see that the very first question up here is, do we have gram-positive cells? And so what we do for our first test is basically just a gram stain. We've talked about gram stains previous in this class. A gram stain's a relatively simple procedure, takes a couple of minutes in order to be able to do it. We end up with a result after that where we end up with either gram-positive or gram-negative cells after that. And what we see is that if we end up with a gram-positive cells, it says they're gram-positive bacteria. The blue box is indicating go to another page with another dichotomous key to help us to identify what that gram-negative or gram-positive bacterium is. If we have a no result, if we have gram-negative cells in here, we want to look at the morphology, we want to look at the shape. And if we have cocci or pleomorphic bacteria, it says go to another dichotomous key on another page in the book. If we end up with rod-shaped bacteria, we go to this one. The question is asking us, are you able to tolerate oxygen? A simple biochemical test that we can assess relatively rapidly. If we get a positive result, it's going to another biochemical test. Are you able to ferment lactose? If we end up with a positive result, we go to yet another biochemical test. Can you use citrate as the sole carbon source? If the answer to that question is no, it goes to another biochemical question. Do you produce gas from glucose? And if we have a yes answer, we can see we've identified our unknown as Escherichia coli. What tests are we running? Well, we are running a gram stain. We are looking at the shape of the cells on our gram stain. We run an oxygen tolerance test. We run a fermentation test using lactose. We run a citrate test, and we run a producing gas from glucose test. And if we end up with a no, yes, 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 no, yes result, we know that our microorganism is Escherichia coli. If we go down this dichotomous key and we end up with a negative result from our producing gas from glucose, our microorganism would be ID'd out as Shigella. Now, the dichotomous key in the previous slide is actually, for me, very easy for me to follow. It's a very intuitive flow chart to be able to come down and identify microorganisms. But we can see that same dichotomous key is presented in outline bullet list form, where our first question is the gram staining, and if we end up with a positive result, we go to a new key, and a negative result directs us to question 2. If we end up with a cocci or pleomorphic bacteria from non-rod-shaped cells, that's telling us to go to a new key, and our answer 3 rod-shaped cells would direct us to question number 3. This dichotomous key has the exact same information that's here in this flowchart form. And the reason that I'm putting this up here and making the distinction between these is that you may see dichotomous keys in both these sort of formats, and we want to be very comfortable as a learning goal from chapter 4 to be able to utilize a dichotomous key like that to be able to identify an unknown microorganism. However, when it comes time in a laboratory report to actually reproduce our own dichotomous keys as part of that laboratory report, it may be useful, it may be easy for you to be able to show it in flowchart form, or if you really don't like doing word art, putting it in outline form like this may actually be easier for you. In either one of these formats, flowchart form or numbered bullet point list form, are appropriate ways to be able to put the information from a dichotomous key. So that's the end of chapter 4. Hopefully from watching these YouTube videos we now have a good appreciation for some of the different strategies that we can use to be able to identify microorganisms. It's of course an essential learning goal from this portion of the course to be very comfortable with some of the tools that we might want to use, and as clinicians, as allied health workers, being able to identify unknown microorganisms is really a key step in the process of being able to move on to getting a beneficial patient outcome.